so let's go ahead and get started. Welcome to the Southern IPM Hour. This one is on an ARDP presentation called Identifying Overwintering and Landscape Features Associated with Stink Bug Risk and Yield Loss in Corn. I think this is going to be super interesting. I'm so excited to hear about it. And we are the Southern IPM Center. We present research issues and programs in integrated pest management from the Southern region through this webinar series. I'm Kayla Watson, the Communication Director for the Southern IPM Center, and we are housed at both NC State and UGA to coordinate IPM across the region. We're also one of 4 regional IPM centers supported by the USDA NIFA Crop Protection and Pest Management Regional Coordination Program. But for the webinar today, this project is an ARDP or Applied Research and Development project that's a part of the Crop Protection and Pest Management Program at USDA NIFA. But today, we're going to hear from Dominic, who is a professor and extension specialist, a university faculty scholar at the Department of Entomology and Plant Pathology at NC State. And Dominic, thanks so much for joining us. I'm very excited to hear about your project.
Yeah, thank you for the opportunity, Kayla. Thank you to the Southern IPM Center for hosting this opportunity. And thank you to the USDA, NIFA, CPPM, ARDP program for funding. Applied research is what my program does, so this is a great fit. Before I tell you about what we did with the funding, I need to start out with a little bit of background and tell you about the system that we're working in. There are 13 species of stink bugs that are pests of corn in the US. They're an increasing problem. We have an invasive species called the brown marmorated stink bug, which maybe some of you have seen in your houses. That's where they like to overwinter. We have a species called the red-banded stink bug, or Pesiodorus gildini in the Mid-South. And we have the Euschistus genus that's widespread in corn across the US. The species that we're mainly going to be talking about today are Euschistus servus, Chinabia, and Nizara viridula. Those are the main species that we have here in North Carolina. They're a pretty big pest in the southern US. You can see that 93% of the yield loss that we have from this pest in corn happens here in the southern US. I've been compiling some yield losses with a bunch of other corn entomologists across the US, and we've broken out what are the top pests by region. And the pest that dominates US agriculture in corn is western and northern corn rootworms. They cause huge losses in corn, primarily in the Midwest. But if you look at the bottom of that chart and you look at the southern region where I'm located, you notice that stink bugs top that list. And then following that are 2 types of mites. And that's primarily because Texas has a lot of, it's a big state, has a lot of corn acres, and that's where most of the mites are. But here in the southern and southeastern US, the stink bugs are really our main insect pest. They can injure the corn in several ways, and it sort of depends on what time of the growing season that they're feeding. When corn is a seedling, if they feed on the corn early enough, they can actually kill the plant. Most often what folks see is the transverse holes through the leaves. You can see that out on the picture on the far left. The stink bug's feeding mechanism is called a stylet. A stylet is like a hypodermic needle. They'll use that to insert it into the plant. They'll eject saliva out of that stylet. That'll help macerate some of the tissue for them to feed. Causes just direct injury to the plant itself from that laceration from the stylet, but also those saliva enzymes degrading plant tissue. And because the corn plant, its leaves are whorled and furled up before they expand, when it does expand, because it's feeding through that circle, what you're left with is transverse holes in the leaves. And you notice some of the nutrient deficiencies, that yellowing on the plant that you see on the left. They're difficult to sample. I've circled the stink bug in the middle. You can see how they really blend in with the plant and the soil. And then sometimes they can cause what's called tillering in seedling corn, where the main part of the plant is there, but it's putting out additional vegetative shoots called tillers, because corn is a grass. And that, of course, can rob yield. Fortunately for corn, the growing point is usually below the soil surface. This graph on the left shows the corn growth stage, depending on how many leaves it has, from V2 to V6. And we've got the soil line there at the 0 graph, and you can see that when corn has 2 leaves, the growing point is actually about 2 centimeters below the soil. But you can see on the picture on the right, the stink bugs can actually, depending on the soil type and how movable it is, can actually insert their head and feed below the soil. And really, the only time we see injury to the plant in the seedling stages is kind of between that V2 and V5 stage. We think once it gets, V6, V7, V8, it no longer causes significant injury to the plant. A seedling injury is not something that's super widespread in our system, and we think that's because of the widespread use of insecticidal seed treatments. Every single corn seed that's planted in the southeastern US comes with a base rate of insecticidal seed treatment on it. We've done studies in my program. This is an example from a publication and a program in Virginia Tech. And you can see the effect that these corn seed treatments have on reducing injury from stink bugs to the corn plants. And you can see that visually from the pictures on the right. Basically, with the addition of the insecticide, when you look at a metric like leaf height, it's controlling the insect, allowing the plant to grow better. And we think this is one of the reasons why we don't see widespread injury in seedling corn. The main type of injury that we do see is expressed once the corn is in the reproductive growth stages. This happens when the insect is able to feed when that primary ear is forming. You can see my finger holding that little salad bar sized ear on the top. What the stink bug is able to do is it's able to actually feed through the stalk of the plant and reach that primary ear as it's forming. That primary area has cells that are not divided yet. The mother cell divides into different daughter cells, and you can imagine if you take out that mother cell, it's no longer able to divide into those daughter cells that are supposed to surround it. And so what you get with is a deformed, misshapen ear. Growers have a lot of different colorful names for it, but probably the most commonly used term is banana ear, because the ear is crooked over like a banana. In addition to causing direct yield loss, they can expose the tip out of the husk, which can introduce pathogens into the corn and cause issues with disease associated with the feeding. And then the final time that the stink bugs can feed in corn is later on in the reproductive stages. This is after the kernels have pollinated, and when they're filling grain, they can directly destroy the kernels themselves, or they can reduce the substance, the grain as it's filling into the corn. And then you can see that they can introduce pathogens as well. You see that starburst on the kernel. Those are mycotoxins that are growing as a result of pathogens that have been transmitted through the feeding of the stink bug. This is a survey that we did a number of years ago. I believe 2018, 2019. We surveyed over 100 corn growers in eastern North Carolina, and we just asked them, what percent of corn area was fed on this last year, or sorry, what percent of corn area was sprayed for stink bugs last year? And then what do you think the cost and losses are to your corn? Look at the black there. Those are growers that are not spraying very much for stink bugs, growers that are not having many losses. But at least in this year, in this region, the majority of growers there fall into one of those colored pieces of the pie. They were either spraying for stink bugs, or they think they're receiving losses and spending money on controlling stink bugs. So in our region, you can see it's an issue, and in some cases, growers were spraying 90 to 100% of their acres for stink bugs. And that was 241 growers that we surveyed. So probably hundreds of thousands of corn acres represented in that survey. The primary way that stink bugs are treated in field corn is by using aerial sprays. These aerial applicators are putting out insecticides at fairly low volumes, anywhere from 1 to 3 gallons in an acre. Growers can pay more to have them put it on at 5 gallons an acre. And in a lot of cases, these aerial sprays are working. We can follow up these sprays in the field, see that the stink bugs are lying on the ground, they're dead. In some cases, they're not working at all. This was a study that we did. Okay, this was a study that we did in the early 2010s, where we compared stink bugs on plants that had been treated aerially. You can see the stink bug numbers in the treated plots in the dashed lines there, untreated in the in the solid line, and you can see there was no significant difference in treatment before or after application. So this is one case where aerial applications clearly are not reaching the stink bugs where we need to be. However, we do know that if we can apply insecticide to the stink bugs and deliver it to where they're located, we can have efficacy. This is a efficacy trial where we sprayed stink bugs at tasseling in corn. The lines in the blue are the numbers at 3 days after treatment. The lines in the red are the numbers at 6 days after treatment. You see the check numbers on the left, a lot of different types of insecticides. We're able to lower the numbers. The insecticides that you see that have the NL behind them, those are insecticides that are not labeled in corn. So they're not permitted for growers to use. And so that you can see that among the insecticides that we were used in this trial, 2 types of pyrethroids were effective for stink bugs, bifenthrin and lambda-cyhalothrin. And the insecticide that we recommend growers use is bifenthrin because, as you can see, it has more of an efficacy on those brown stink bugs than other types of pyrethroids. So growers are essentially relying on a single mode of action. We don't have a lot of other options in corn. There's nothing that they can really rotate to that's labeled that's also effective. So we have to try to increase IPM in corn in other ways other than rotating insecticides. We know a little bit about their pattern of colonization, not only in corn, but in other crops. This was a really nice study that was done 2009, 2010 in southern Georgia. And they looked across their cropping system and looked at the number of stink bugs that they found in different crops. You can see corn there in the line that is a solid circle, cotton in the line there that's a open circle, peanuts that are represented by the solid triangle, and soybeans that are represented from the open triangle. And notice that corn is one of the first crops that's colonized in that system during the year. Where I'm located in the southeastern US, we have another very important crop that's wheat. And so I think if I were to do this study in my system, I might see something like this here, which is a peak in wheat before they move to corn. You can look across the landscape and notice areas where there are vast acreages of wheat next to corn. If these stink bugs are building up in wheat, oftentimes when growers harvest the wheat, they then move to the next available host in the landscape. If corn's next to that wheat field, that's the crop that those insects move to. But the thing I want to highlight with these 4 pictures is not every wheat to corn interface is the same. Sometimes there's on the top left here, for example, an empty ditch, or there may be a road, or there may be a ditch, but it's grown up in weeds, or maybe you have a ditch that has a trees as a barrier, or maybe that ditch has water in it. So even though there's wheat next to corn, their movement is not always predictable. It depends on how that interface looks, where they are in terms of development, and what other available hosts there are to feed in the environment. However, we have shown that they do in fact move from wheat to corn following harvest. These are stink bug numbers in corn right next to a wheat field. Anywhere we have that black circle, that's where the stink bug numbers are heaviest. So on the picture on the left, you can see that 4 days following wheat harvest, we were picking up a ton of stink bugs in corn next to that wheat field and not a lot into the cornfield. And then 8 days following that harvest, you could see that movement of stink bugs into the corn from the adjacent wheat that had been harvested. So crops in the landscape are really important. Want to turn now to corn itself, that second crop in the system that is fed on by stink bugs in the southeast. We've already talked a little bit about feeding on seedlings in corn. We've talked a little bit about that injury that occurs to the primary ear as it's developing. When the corn plant's in the vegetative growth stages, prior to tasseling, it has a number of different primordial ear buds. Those occur at each and every node on the plant, and you can see that represented in this picture here. If you can see those, let me get this little laser pointer here. Hopefully you can see these little primary ear buds that are formed at every single node. The corn plant around V5 decides, and I'm using air quotes here, decides which one of those is going to be the primary ear. That's the ear that it's going to put all those resources into to form its yield, and that's what we're trying to avoid the stink bugs feeding on. This is the picture here on the right shows the primary ear as it's forming. It forms from that very small primordial earbud into an ear leaf sheath and a more visible ear. And it happens very rapidly, anywhere from 1 to 2 weeks prior to tasseling, just in development. And so really growers have a very narrow window with which to target that stink bug to prevent that primary ear damage and that stink bug from feeding on it. We worked a lot on developing scouting recommendations for growers. We published a number of studies. I had an excellent PhD student who's now a postdoc at the University of Georgia. He's on the job market. He's excellent. Contact me if you're looking for somebody. But he did a lot of good work looking at where the stink bug was located on the plant, how to scout it. And one of the things he found is that once we near the reproductive growth stages, those stink bugs tend to congregate near to the middle of the plant, wherever that primary ear is forming. And so what we recommend growers do, and scouts and consultants, is define where the primary ear is forming by pulling back the leaves until they find that primary ear leaf sheath growing there. You can see it represented by an arrow. And based on that, we've developed a sequential stop sampling plan with economic thresholds based on different growing stages of the season. And based on that sampling work that that student did, we found that growers didn't need to sample the whole plant. They could only sample part of that plant and save themselves on average 60% of sampling time. Now, this work was also based on some cage work that we did in collaboration with Virginia Tech. And so we have very good up-to-date thresholds for stink bugs in corn, thanks to this work that was done. One of the additional things that makes stink bugs difficult to scout in corn is they tend to be sporadically distributed throughout the field. This is a study in southern Georgia that shows the kernels that were injured by stink bugs. And you notice that a lot of them tend to be near the edge of the field, and they tend to be sporadically distributed. So growers, scouts, consultants really don't like this. They'd like to be able to enter the field in 1 or 2 spots and make a decision on whether or not to treat that field. But unfortunately, what we have to tell them is you got to scout all around the different edges and sometimes even into the middle to know if you have stink bugs. You can't just make a uniform decision because they're so sporadically distributed. And so this just kind of makes scouting a burden for growers. One of the things we started doing is looking at different IPM tactics we could use in the landscape. As I mentioned, we only have that single reactive measure to control them, which is a single mode of action of insecticides. One of the things we did is work with a very large grower in eastern North Carolina that primarily farms corn and soybeans. He farms very large acreages, and he flip-flops his rotation every year. And he was having problems with stink bugs in his corn where he was growing soybean the year prior. And one of the things he told me is that the stink bugs were coming out of the edges and feeding on his corn. And this is just a picture of what one of those edges looks like. You notice how uniform the edges are in this field. It's a very uniform environment, a very simple ecological system, if you will, to work in, and one that we thought we could have some effectiveness in helping him control the insect. So we did a lot of work on the field edges themselves because we found stink bugs feeding on those canal borders and things in weeds, and we were able to characterize some of the stink bugs that were in weeds. One of the things that we found in those weeds is that stink bugs tended to be more prevalent in weeds when soybeans were planted to soybean the year prior compared to corn. You can see that here on the graph on the left, how many more stink bugs were in weeds when soybean was planted the year following compared to corn. But the effect was not consistent. So you see here our data from 2017. We didn't find the same thing. Well, we did find that effect in 2016, and we thought, well, what if we made manipulations to the weedy border? What if we mowed the weeds? What if we applied herbicide to the weeds? If we destroy the weeds, can we influence injury into the corn? At least in our case, the answer was not really. So here's where we have examples of mowing, yes or no, herbicide, yes or no, and whether we were looking at stink bug numbers in corn or the stink bug numbers in weeds, we could modestly affect the stink bug numbers in weeds. That's the graphs that you see here on the right, but we essentially had no effect on stink bug injury in the corn. And it seemed like there was some other source of stink bugs in the environment other than weeds. They were there, but clearly something was influencing the stink bug numbers in corn beyond weeds. And to be clear, there were no other crops surrounding this farm. I showed you that picture of the border, surrounded by woods. The nearest crops were maybe a mile away. Well, we did notice that stink bugs kind of dispersed differently depending on what weeds they came off of. This is where we had attached some stink bugs to a flight mill. When they came off weeds early season, they seemed to fly a lot more than off some of these crops. But to be clear, this is confounded by effects in the season. You know, we were pulling them off weeds early and comparing them to weeds late. You know, maybe they just fly more because it's spring versus fall. But we had some inklings that, yeah, you know, maybe depending on what host they come on, maybe that's influencing dispersal, and maybe that's having an influence on whether or not we see them in this environment. Well, one of the things this student did is he thought, well, maybe they're coming out of the woods and feeding on things in the woods before they move to the weeds or the corn. These woods were very uniform environments, primarily plantations of loblolly pine, managed underneath with no weeds underneath, just pines and pine needles underneath there. One of the things the student did was to cage stink bugs on those pines. And this is the survival in days after caging on the x-axis of adult brown stink bugs that were caged either on a dead branch of pine, a juniper plant, or the needles of a loblolly pine. And you notice that they could some of them could survive for very, very long time periods just on those needles of that loblolly pine. So it looks like in this environment, what may be happening is these stink bugs are emerging from overwintering sites, feeding on whatever they can find in the environment. It could even be pine needles. It could be weeds, whatever. When the corn's available, they can then feed on corn. Our hypothesis based on this system was that stink bugs would follow a predictable pattern of crop sequences throughout the year. They could use crops during the autumn as sources, maybe things like soybean or cotton. They could then move to overwintering sites, and it could depend on the forest type for how well they could survive. Survival might be different in a forest that's dominated by conifers, might be different in a forest dominated by deciduous trees, or might be different in a scrubby environment. And then that may influence stink bugs as they moved into corn. Based on that, we moved to the objectives that we had for this ARDP grant. We wanted to look at these different 7 objectives. I will not be speaking about the first 3 objectives today. I'm going to talk about them at the end of the presentation. We're going to skip right over them now because this is ongoing work. We were not able to do these 2 objectives. It just proved too unwieldy. We wanted to estimate stink bug injury in fields and get yields from the growers, but it was just too difficult to work with the number of growers that we had to get that information. And so because we were not able to do those 2 objectives, we pivoted and looked at the effect of rotation. So I'm going to talk about these first 2 objectives now, and then pivot and talk about the 2 objectives that we added to make it for the 2 that we couldn't do later. And the first 2 objectives we're going to talk about now are characterizing the overwintering habitats based on categorizing the host plants in the forest, and then measuring the colonization into corn next to those overwintering habitats. So to achieve this objective, we sampled stink bugs in corn in 2020 and 2021. We did this in 2 distinct ecoregions in North Carolina, an area that was in the central coastal plain, which is categorized by urban areas, woods with lots of overwintering habitats, smaller fields, and relatively diverse crops compared to an area that's called the Blacklands, where it was much larger field size, more uniform, lower crop diversity. We sampled 11 fields from the coastal plain in 2020, 17 in 2021, and then from the Blacklands, we sampled 12 fields in 2020 and 14 in 21. From those fields, we sampled 800 corn plants weekly throughout the season, and this is just a graph that shows the average number of stink bugs per 800 corn plants per sampling visit across the season. And if we compare those different ecoregions, we notice that there were fewer stink bugs overall in the Blacklands relative to the coastal plain, the central coastal plain. And that in 2021, our numbers were a little bit higher in the Blacklands compared to the central coastal plains, very low in 2020 in the Blacklands. If we look at the number of fields that exceeded threshold, no fields exceeded threshold during the seedling stages. And remember, I told you earlier that we think that that's because these insects are pretty well controlled with the insecticidal seed treatments that growers are using. We did find fields that exceeded threshold, however, during those pre-tassel stages. It's causing that banana ear that I told you about earlier. None of those fields were in the Blacklands. Nearly half of the fields in the central coastal plain in 2020 exceeded the economic threshold. 3 out of 17 exceeded the economic threshold in 2021. And then once we got into the later reproductive stages, R1 to R3, sorry, not later reproductive stages, they're still early, but later on in this study, only a single field exceeded threshold, and that was in the central coastal plain. So you can see that we saw distinct regional differences about where stink bugs were, even within the same state, pretty close in terms of driving distances. If we just break that data down and look at the central coastal plain itself, we saw a pattern of distinct colonization by stink bugs as the corn progressed throughout the growing season. This is the average stink bug numbers per field in the central coastal plain in 2021. That's the blue line. And then in 2022, that's the red line. And we've now broken it out where we've taken a field average by growth stage. Anything you see here under this dotted line, that's the vegetative growth stages. After the corn is vegetative, of course, it enters the reproductive growth staging and tassel, which is R1. And you notice that those stink bugs are really colonizing the fields in those later vegetative growth stages, which unfortunately is when that plant is starting to form that primary ear. And this is the time where growers really need to focus on controlling those stink bugs in those cornfields. So just to reiterate, we did find that stink bugs in corn were associated with locations. It was highly important whether they were in the central coastal plain or Blacklands. But what about those overwintering sites? Well, we went to each overwintering site and we carefully measured with transects what species there were of all different herbaceous vegetation, woody vegetation. We measured thickness of the trees. We measured the amount of duff that was on the soil. We measured everything we could just to characterize those overwintering sites next to those cornfields. We took all that information and we put it into a stepwise regression. Stepwise regression is nice because it lets you determine which of those variables that we measured is going to be important in predicting stink bug abundance into the cornfields. And based on all that information we took, we had to break it out in terms of woody vegetation versus herbaceous vegetation. So this particular stepwise regression is putting all the woody vegetation information into this analysis. And what popped out at us was percent duff cover and percent duff depth. If you don't know what duff is, it's the stuff that's left from the trees and vegetation during the fall after it's dropped its needles and leaves. And essentially what the regression was telling us is when you look at woody vegetation, it doesn't matter what trees are out there, how many there are, how big they are, how much duff you have and how deep that duff is was predictive of whether or not stink bugs would be in that cornfield. Here's the stepwise regression with the non-woody vegetation, the herbaceous vegetation. And in this analysis as well, the same variables popped out at us. Didn't matter what vegetation you had, percent duff cover and percent duff depth was important in predicting the stink bugs that were in adjacent corn trees. This was also confirmed with a principal components analysis on those loadings on those first few factors. Duff cover and duff depth, which you see down here, popped out as important for the woody analysis. And then for the non-woody analysis with the herbaceous vegetation, percent duff and percent coverage were very important for predicting whether or not stink bugs would be in that cornfield. So in addition to finding that location was important, we found that duff was important. Indeed, overwintering sites are important. It doesn't really matter what's in those overwintering sites. It just matters how much of that duff there are. And that makes sense because that's probably where those stink bugs are overwintering. They're probably going underneath that duff. They overwinter as adults, and that duff, probably the more of it is and the thicker of it is, probably buffers them against extreme winter temperatures and provides them an opportunity to better survive. Okay, I told you that I would talk about the objectives that we had to pivot on. That's what we're going to talk about now. And we really wanted to focus on some of these crops in the season that may be influencing corn during the next year. You'll remember that we found that in weedy borders following soybeans versus corn, at least in 2016, there were a lot more stink bugs in soybeans. And so maybe what crop you have the year prior predicts how many stink bugs are going to be in corn the next year. Now, this is bolstered by some additional studies that were done in southern Georgia. They found that soybeans were the most important driver of stink bugs in the system because, as you can see from this graph here, all these crops are gone from the field in late fall. The only crop that's available to stink bugs for them to feed on that's cultivated is soybeans, and that's where they tend to have their second generation of the year. So soybeans are potentially very important to stink bugs in the system for corn the next year. So what we did is a 2-year study. We identified fields in the central coastal plain of North Carolina that were either planted to cotton or soybean the previous year and that were planted to corn this year. So we went out and sampled those fields for stink bugs, and we wanted to do a very simple comparison. Would there be more stink bugs in corn if soybeans were planted the prior year or if cotton was planted the prior year? Here's the data just looking at peak stink bug averages. So that's just the day that there were the most stink bugs in corn. Was there a difference between cotton and soybean? This is a p-value here that's not significant. There's a trend to more stink bugs in corn following soybeans, but not significantly more. We saw similar patterns when we looked at the number of stink bugs in the pre-reproductive stage of corn. So that's that time period when that primary ear is forming. Very similar results. A trend for more stink bugs in corn following soybeans, but not statistically significant. And then finally, looking at the post-reproductive stages, tasseling, grain filling, and whatnot, the exact same results. There's a trend for more stink bugs in corn following soybeans versus cotton, but the results were not statistically significant. So this was a good result in that it seems like the system in the coastal plain of North Carolina is not matching southern Georgia. Soybeans are not super predictive of stink bugs in corn. You can have them whether it's soybeans or cotton the previous year. But it's a little bit disheartening because we were hoping that we would see a very distinct trend there that would allow us to advise growers and give them a predictive risk tool and say, hey, if you've got corn planted following soybeans, you're at X percentage of risk compared to if you're planting cotton. And because there's really no significant difference, we can't see that. I do feel like we know a lot about the system. We can kind of predict some of where they are. We know how to kill them. But really, we need to do some additional things to tie a bow on this. And that's the additional objectives that we weren't able to accomplish in our 3-year funding period. We really need to do some more work on measuring stink bugs in crops the year previous to corn. My program has a collaborative project with my research counterpart at NC State, Anders Huseth. We're doing a lot of work sampling soybeans and characterizing stink bugs there. with some funding outside of this grant. We would eventually like to create a risk map for growers. In order to do that, we need to have very good measurements of what's causing those stink bugs to be there. Right now, the only thing we can really point to is that duff metric that I showed you. So maybe we can get some characteristics of that to help growers. I do feel like we were able to document some baselines of risk management for stink bugs to growers. For example, we were able to show growers in the Blacklands region of North Carolina that stink bugs didn't exceed threshold in a couple of years, whereas in the coastal plain they did. And so you're going to have distinct regional differences, which really emphasizes the need for careful scouting of each field. And we're already disseminating this information to stakeholders. I just presented this information to growers yesterday in the southern part of the state. I think there were over 70 growers there. Talked about it earlier in the week, and the student that worked on this project presented on it as well. Would like to conclude by thanking our funding agencies. The corn growers of North Carolina funded this work, USDA NIFA, and special thanks to the Southern IPM Center, all the graduate students that worked on this, Arun Babu, the talented scientist I mentioned that's a postdoc in Georgia, Kevin Orda, who's the current PhD student working on this, and then all the student workers and whatnot that helped us. And Kayla, if I have time for questions, I'll take them.
Sure. So I don't see any just yet. But my question to you is, what were you most surprised about with your study? Is there something that you were going in and you're super sure about and then it ended up not being true, or anything that was particularly surprising?
I was surprised about thank you for the question. I was surprised about two things. My first surprise was the regional difference. And I guess I wasn't surprised that we had a regional difference. I was surprised that we found so few stink bugs in the Blacklands region of North Carolina, because this has traditionally been the region that we think of as our stink bug hotspot. And we now think that Central Coastal Plains is the stink bug hotspot. And we keep going there year after year, and that's where we find the stink bugs. So that was surprising to me. And the second surprise was the lack of really strong differences in the rotational study, because they saw such strong differences in southern Georgia. And I think the difference might be they did some really careful repeated studies on a relatively narrow number of fields in their landscape, whereas we looked at a ton of fields across a wider landscape. And so I think that may be some of the difference there, is just the scale at which we were looking at things. But it's good to know that there's differences.
Yeah, definitely. For me, it's interesting that you had different results than folks in Georgia, because you wouldn't think it's pretty close, right? Still the southern United States, the southeast, but they are a little bit different climates. So it would make sense that there's a difference, but it's also a little surprising to me that it's that different.
Yeah.
Let's see. Okay, so we do have a question now. And they say they're looking at the threshold for stink bugs. How do you make the decision to spray when you have other pests like western and southern corn rootworms?
Yeah, thank you for the question. I would say from a southeastern perspective, there's probably very few acres where those two pests are going to overlap. We have issues with western corn rootworm in the mountains where folks have dairy operations, they're growing corn for silage, and they're not able to rotate because of the economics of scale. So they just are growing corn on corn on corn. And of course, western corn rootworm, that's how it thrives, is lack of rotation. And the stink bugs that we work on in the coastal plain are not prevalent as you go to that part of the state. Brown marmorated stink bug might be prevalent, so there may be some overlap there. And then in the Midwest, the western corn rootworm tends to dominate. Usually stink bugs aren't exceeding threshold. But I would, just to answer the question directly, if that question were posed to me, if I were given that situation, I would probably ask a lot of questions about the specific nature of the field. And I would say if they exceeded threshold for stink bugs, to go ahead and treat, and then to continuing monitoring for western corn rootworm. And I know they're using sticky traps and whatnot to make adult sprays there, and hopefully they can have those sprays overlap. But if not, they may have to make a dedicated spray for the adults, themselves. So I think you have to manage each pest independently and just hope that you get some overlap on them.
Thank you for that answer. I really appreciate it. And don't forget, let's see. Oh, I think this is a follow-up. For the influence of the previous crops you mentioned, did you also consider nearby overwintering sites like Duff, as you mentioned in part of the study?
Yeah, thank you for the question. So you'll remember in that second study, we looked at the influence of previous crops rotation, whether it's soybean or cotton, on whether stink bugs were in that cornfield or not. And we did not characterize overwintering sites in that study because it took a lot of work. And by the time we knew duff was important, which actually doesn't take a lot of work, you can just go in the overwintering sites, throw a ring down basically and make an estimate of how much duff there and take a ruler and see how deep it is. That would be a pretty easy one to do. By the time we had that answer, we'd already sampled the field. So I appreciate the question. Maybe some future work we should do is to layer some duff measurements on top of it.
So a question that came to my mind as you were speaking, Dominic, you talked about not having very many IPM practice tools to use. You've got being able to rotate the modes of action right now with the insecticides, but you're hoping to be able to use or just add more of those practices to your toolbox. In a perfect world, what other tools would you like to be able to have when managing these stink bugs?
Yeah, thanks for the question. To be clear, I think corn growers do use some IPM principles for pests. They're planting on time, they're using good fertility, they're managing their weeds. All these things help keep pest insects down. But in terms of reactive tools we have for stink bugs, we're limited, right? So we have some of the basic principles of IPM, which are to identify your pest. They're able to identify the stink bugs. We have economic thresholds. We have good sampling methods. What we're really lacking are some of those higher order IPM principles where we're not able to rotate insecticide classes, for example, or we're not able to use less broad spectrum insecticides that might preserve some beneficials, for example. And so it would be really nice to have some more hard technologies, I'll call them, like some more selective insecticides that are registered in corn that are effective on stink bugs. Unfortunately, of insecticides that are commercially available, even in other crops that's not available. Most of the types of insecticides that we have to use for stink bugs are broad spectrum, and we just don't have a whole lot of other options. So I'd love it if we had other options for stink bugs in corn. We tried some of the cultural practices. I showed you that study where we mowed weeds on the ditch bank. We didn't have an effect. So
Right.
I think the answer is probably just better insecticides at this point.
Gotcha. I was just curious about what those practices you were missing or hoping to have more, and that definitely makes sense, especially when you have those broad spectrum, as you said. You could be also targeting the beneficials, and definitely don't want that. So yeah,
One thing I should mention as well is I think we've had a lot of success in the region because the way growers thought they were managing stink bugs in corn before was they were tank mixing an insecticide with their fungicide, and this application typically went on at tasseling. And I showed you most of those stink bugs are colonizing the plant before tassel, and they're causing the injury to the plant before tasseling. And so we've gotten the message across to growers that you need a scout. Growers are in some regions are actually paying folks to scout their corn, and they're putting on dedicated applications. One thing we could use more to improve IPM is like high clearance sprayers, because in that central coastal plain area I showed you, it's difficult to get on aerial applications. And so if they had sprayers that could get over top of the corn and apply the insecticide better, they'd be better off.
That makes sense to me. So just one last question to end your talk today, because I'm not seeing any more in the Q&A box right now. First, thank you so much for your presentation. I think it's great information and definitely some questions that you didn't get answers to, but it's always good when you have at least one research question where you can say, yes, exclamation point. That's amazing. So I'm very happy for you for that.
It wouldn't be research if we knew the answer, right?
Right. Yeah. And then you couldn't do more research. So, but at least you did have that one yes. So when people are leaving your talk today, what is the main takeaway that you want the attendees to remember from your talk?
Main takeaway. I would say that we can manage stink bugs in corn. We need to be careful about timing, and we need to think about things outside of the system, outside of the field, right? We need to take the biology of the organism and the system into account. And that's really what we were trying to get at with this overwintering study here, is we were thinking beyond just that field that year and how that might influence pest management practices that year and into the future.
Well, thank you so much for that. And we do have the National Program Leader for USDA, NIFA, CPPM, Emmanuel, on the call, and he says, great work. So he's giving you a round of applause.
What an honor. Thank you very much.
So thank you again, Dominic, for your presentation today. You just know that when you close your window out today, there's going to be a quick pop-up survey after the webinar ends. It's just 5 questions. It just helps us guide the future of our webinars, making sure that we're hitting the information that you need to know. Our next webinar is coming up actually next week, so next Wednesday. And this one is actually on cotton leaf roll dwarf virus. So one of the crops that Dominic was talking about today, I know he talked about corn, but this one would be more cotton focused. And it's on epidemiology and management of CLRDB. So that's going to be with PI Alana Jacobson. It's another ARDP presentation, and that will be next Wednesday at 1 o'clock. So we hope you can join us there. Again, a link to a recording of this presentation will be sent to your email and will be available on our website after the webinar. So if you're talking about it with a friend and you want to share this information, you can definitely do that and you'll have access to that video. If you want to find out more information about the Southern IPM Center, you can always visit us at southernipm.org, and we'll see you next time. Thank you so much.